Good morning, afternoon, or evening. Thank you for joining my presentation. I'm Wissam. I'm a second-year PhD student at Ghent University in Belgium at the Department of Data Analysis and Mathematical Modeling. With this presentation, I hope to elaborate a bit on the main idea of what I'm working on during my PhD. My PhD project is part of a bigger interdisciplinary project in collaboration with the Marine Biology Department at our university. In this bigger project, the main aim is to assess the ability of marine ecosystems to acclimate and adapt to rapid changes in their environment. The major contribution of our research unit lies then in the development of spatiotemporal models to help answer questions related to this main objective. With ongoing rapid changes in the environment, populations around the globe are severely impacted. A set of example questions that are frequently asked are then, for example, which populations will be most affected, in what way will they be affected, and at which rate will these effects take place. So solving these questions can also guide policymakers in taking appropriate and efficient measures. But of course, these are no easy questions to answer, as the relationship between a population's performance and its environment is complex in nature. Predictive ecological models that account for the complexities in this relationship will therefore be important to formulate answers to these questions. Understanding population responses to their environment can nicely be summarized by this framework proposed by Johnson et al. Physiology is a key and important element linking environment with the performance of population through individual mechanisms. So this has led, for example, to DEB-IBM providing, DEB-IBM being dynamic energy-based budget individual-based models, which are population models using dynamic energy budget models to govern the physiology of individuals. So these models provide a nice basis to model populations facing climatic changes. However, other mechanisms such as behavior and evolution also need to be integrated for these models to account for the other complexities. So more specifically, work on integrating the impact of eco-evolutionary dynamics in these models remains scarce. My recent work mostly focuses on on the integration of evolutionary models in such physiologically based population models. Key questions that I will try to elaborate on in this presentation to explain my work a bit further are: how could we extend DEB-IBM for eco-evolutionary modeling purposes? Where do the challenges lie for such an approach? And what are the potential benefits? So solving these questions can then further help in answering the questions that we're looking at in the general PhD project as to how populations can adapt and acclimate to environmental changes and what the impact of eco-evolutionary dynamics exactly are to these responses. So given this background, we proposed an implementation of this extension to the generic DEB-IBM by accounting for eco-evolutionary dynamics. And here is a schematic overview of this extension. But conceptually, of course, the general model remains similar to the generic DEB-IBM. So we have the dynamic energy budget model that governs the organism's energy budget in function of temperature and food, which results then in the expression of its life history, which is then extrapolated towards the population dynamics in the IBM, in the individual-based model, by explicitly keeping track of important events such as births and deaths. The major difference here, however, is the addition of an inheritance submodel that takes care of passing on genes from the parent to the offspring generation and expresses also the genes into phenotypes. The question, though, still remains then: what exactly are genes and genotypes in the context of a DEB-IBM? For this, we have considered genotypes, genes, and phenotypes to be modeled based on the concepts from the field of quantitative genetics. So we start with the assumption that variability in any of the life history statistics does not necessarily fully originate from measurement and observational errors, but that they also might have a biological origin and potentially a genetic one. Given the fact then that that model structure as well as the conversion and auxiliary parameters remain unvaried, you can then assume that from the mapping between the parameters and the data, that parameters can also vary between individuals of the same species, and that some of the variance can also be genetic. In further illustrations of the model implementation, we considered the case where area-specific maximum assimilation rate, which is a DEB parameter, also shortened to PAM, is assumed to be variable in a population, an approach that is also popularly used in DEB-IBMs to introduce individual variability in a population. Using this assumption, quantitative genetics then describe these traits as the sum of the genotypic and environmental value. Both these values are sampled from a normal distribution with a specific mean and variance. The sum of their variances then results in the total phenotypic variance in the population. The heritability of the trait in the population is then the proportion of the genotypic variance, sigma g, in the total phenotypic variance, sigma p. And this heritability parameter forms as a key parameter for the inheritance submodel. So now that we described what we consider to be genes and genotypes in a DEB-IBM context, inheritance can then be modeled. And also from the field of quantitative genetics, inheritance is described inheritance can be described using the infinitesimal model, which describes how traits are passed on from parents to offspring. And this implementation assumes the simplest case, which is asexual reproduction with clonal inheritance. And from this assumption, then the simple rule follows that the value of the genotype is integrally passed from parent to offspring, while a new independent environmental effect is sampled to generate a new phenotype. And the bigger the heritability, the smaller the effect of the environment will be on the eventual phenotype of the offspring. To then illustrate this implementation for the extension of DEB-IBM, I will present some preliminary results of simulations using the DEB parameters for Daphnia magna, a water flea, taken from the AddMyPet database. Two scenarios are simulated in which all the parameters are identical, except for the heritability of the focal trait, which is in this case PAM, or area-specific maximum assimilation rate. And the heritability is varied between 0 and 0.5. So these simulations show the following preliminary results with regards to the eco-evolutionary dynamics. The plots in the blue colors denote the scenarios without heritability, while the orange color points towards a scenario with 0.5 heritability. So if you focus on the plots on the right, we can notice the impact of ecological processes on evolutionary dynamics. The distribution of PAM in generation 0 is log normal with a mean and a certain specific standard deviation. While 2 generations later, this distribution also remains quite similar. However, in the case of heritability being 0.5, we can see that after 2 generations, this distribution seems to shift and change a bit towards higher deviations from the reference value of PAM. You can then see that ecological processes emerging from this DEB-IBM can drive evolutionary processes within this implementation. On the left, we can see how evolutionary processes affect ecological dynamics. And in this case, the population dynamics in the scenario with no heritability seems to be less performant, showing a lower growth rate and lower equilibrium abundance than the scenario with higher heritability. These results therefore show the emerging eco-evolutionary dynamics from the extended DEB-IBM. Moreover, another interesting preliminary result showing up from these simulations is the emergence of fitness gradients that can change over time. Usually, eco-evolutionary models operate with an a priori assumed function describing fitness in relation to the trait of interest. However, if we can see here, the plot on the left, for example, shows relative fitness, which is defined here as the lifetime fecundity of an individual scaled by the maximum lifetime fecundity within the generation, correlates how this relative fitness correlates with PAM. And in generation 0, this shows a rather linear correlation with higher PAMs, meaning higher relative fitness. However, on the plot on the right, the same is shown but for 2 generations later, and we can see here that this relationship is not the same anymore, and that higher PAMs not necessarily mean higher relative fitnesses. So this actually highlights sort of the advantage of DEB-IBMs as these fitness gradients emerge from the structure of the DEB model within its population context, which is defined by the individual-based model, instead of having to be imposed upon using an a priori function for fitness. And even though these preliminary results suggest interesting applications for the eco-evolutionary DEB-IBM, linking the model with data remains, of course, challenging and a big bottleneck. Estimating individual variability and heritability of DEB parameters from datasets are methodologically very challenging, requiring probably elaborate experimental setups and advanced statistical methods. Recent work within the DEB community also increasingly revealed this importance of estimating interspecific variability in DEB parameters. But for this purpose, eco-evolutionary DEB-IBMs might actually provide an interesting tool when combined with a pattern-oriented modeling approach. This approach focuses on the use of multiple patterns at multiple levels of biological organization to elaborate and validate model outputs and model structure. But for example, by formulating alternative hypotheses on the underlying genetic architecture and variability of DEB parameters, eco-evolutionary DEB-IBMs can allow testing these hypotheses against multiple patterns, for example here the population dynamics and trait distributions, which can help the investigation of heritability and variability in DEB parameters. But for the moment, I'm still refining the methodology and conducting extensive simulation experiments to further know what's sort of happening under the hood of the implemented eco-evolutionary DEB-IBM. But afterwards, the idea is then to apply this model to a marine nematode using both individual level and population level data gathered by collaborators of the project. And then questions such as how do eco-evolutionary dynamics affect nematode population responses to changing temperatures will be more of the focus. And then further down the line, the model development cycle can continue with additional modifications to consider increasingly complex behavior, species interactions, or extra genetic mechanisms. To summarize the main points of the idea proposed here, DEB-IBM has the benefit of allowing dynamic fitness gradients to emerge. Using a quantitative genetic approach for inheritance of DEB parameters can then take advantage of this to simulate eco-evolutionary dynamics. A big challenge still remains, big challenge to use this model for questions related to the impact of eco-evolutionary dynamics still remains the estimation of individual variability and heritability of DEB parameters. But combining eco-evo DEB-IBMs with a pattern-oriented modeling approach can result in a tool that may actually be able to help in tackling these challenges in parallel with the experimental and statistical approaches. I hope you enjoyed the presentation and found it interesting. Thanks for listening, and I'm looking forward to your questions and comments.